Hello everyone. There have been some amazing talks so far today, so it's a real privilege to be here. So thank you, Alison and Anders, for inviting me and making this happen. So a bit about me to start with. My name is Barry Bentley. I've just started the Bioengineering Research Group at Cardiff Metropolitan University. I'm also Deputy Director of the Robotics Centre there. And then from next year, I'm going to be a Fulbright Scholar of Bioengineering at Harvard Medical School, where I'll be working on cryopreservation. Before that, though, I was a worm biologist, and I was interested in the question of extrasynaptic interaction between neurons. So that's what my one-pager is on, and that's what I'm going to talk about now, and why I think this is something that needs to be considered when we're talking about whole brain emulation. So going back to the traditional model of the nervous system, we have the brain, it's composed of neurons, we have synapses between them, and then there's some neurotransmission. And for a long time, that's been considered to be the predominant mode of interaction. And then we have some things on the side that probably don't matter too much. So glial cells have been mentioned, extrasynaptic interactions, neuromodulation, extracellular vesicles. But those have been sidelined and considered to be a kind of secondary, unimportant point. I want to suggest that those elements that we've largely ignored are a lot more important than we've previously thought. And I'll go over two bits of evidence why I think that's the case. So recently, past few years, we've been able to do whole animal functional neural imaging in C. elegans. And one of the things that's come out of that is that we've found that the functional correlation between neurons isn't well correlated with the synaptic connectivity. So if you have two neurons that, where the activity of one is correlated with the other, in only about 1% of those cases will there be a synaptic connection between them, suggesting that there's something else going on. Now, that might be some other exotic mechanism, or it could be any number of things. One potential thing, though, is extrasynaptic interactions between them. And in parallel, supporting that, we know from molecular studies where we've looked at the distribution of neurotransmitters and receptors in C. elegans, we find that the majority of receptors for neurotransmitters are not postsynaptic to cells that produce the corresponding neurotransmitter. So if you look at the distribution of GABA receptors, only about 10% of neurons that have a GABA receptor receive a synapse from a neuron that produces GABA. And the case is largely the same for serotonin, for tyramine, for dopamine, octopamine, and then there are hundreds of neuropeptides as well, where consistently we find that the receptors are not postsynaptic of neurons that produce the ligands. So that's really all I have to say. There's evidence to suggest that non-synaptic interactions are a lot more important than we thought. So this is something I want to raise awareness of, as something that we should probably consider. Thank you.